Great. So welcome everybody to the top 5 design tips for better publication figures. This is a private webinar for TAMU. They are going to cover a wide variety of subtopics, but we've tried to distill it down to 5 basic concepts so you can kind of wrap your head around it. And publication figures are, of course, a little bit different from, say, making figures for a presentation, for a poster, even for a graphical abstract, for example. So we'll go through what those considerations are together.
So at TAMU, we do have a special portal page called, and the portal link is to the top left right there. It's app.biorender.com/portal/TAMU. And all you have to do is just log in using that button that says login now on the right side of the page using your university credentials, and you'll basically get access to free BioRender Premium.
Great. Thanks, Fran. Great. Okay. Well, with that, let's go on to the webinar itself. And so what we'll cover today is sort of 5, again, easy to use tips for better publication figures. We will incorporate with live sort of demos to put some of that theory into practice. And then we'll try to leave time for Q&A at the end, but also, again, we'll go through the webinar answering your questions as they come along. So a little bit about ourselves, I guess, at least the 2 of us that'll be presenting today. My name is Shiz, if we haven't met already, one of the co-founders of BioRender, and my background is in medical and biological illustration.
So hello, everyone. I'm Joseph Zeppa. I'm a new scientific solutions consultant / customer success manager here at BioRender. My academic background is I did a BMSc, Bachelor of Medical Science, and my PhD at the University of Western Ontario in London, Ontario, Canada, specializing in microbiology and immunology, more specifically group A streptococcal pathogenesis. And then just after that, I took my skills to the University of Pittsburgh, where I studied tuberculosis and the immune response to tuberculosis in a non-human primate model. And then it was there actually where I discovered BioRender, how awesome and how easy to use it was to generate these beautiful scientific figures. And I couldn't resist but apply for a position and was fortunate enough to get one. So I'm really excited today to show you how to make some amazing figures, more specifically for publications here. But of course, I feel like this can broadly apply to all of your uses. And I see in the poll that actually quite a few of you had never used BioRender before. So definitely we're going to cover a little bit more about what BioRender is. So it's an online application tool that essentially allows you to drag and drop icons or customize one of our pre-rendered templates to help you build your most beautiful scientific images yet. So it's an online program for creating high quality scientific images. And of course, we've kind of created this for scientists in that you need essentially no art background. So of course, this is really specific for industry, academia, students, healthcare professionals, and really scientific communicators in general. And of course, very important is that little to no art background and little to no time. It's very quick and easy. As you can see, you can just drag and drop images here as our GIF shows. And on the next slide, we'll kind of outline just a couple of our more common use applications for BioRender. But of course, the sky is the limit because our goal here is to give every scientist the tool they need to present their data. So again, here we're going to be focusing on publications and presentations, but you can use this in things like lab meetings, when you're doing your research proposals, if you're building a poster or an oral presentation. Of course, even if you're in the lab all the time and not necessarily presenting a lot, you can really use our tool to build some awesome protocol outlines as well that helps with all of the people in your lab that may be more visual learners. So really, and again, the sky is the limit here. However you think you can use BioRender, definitely we encourage you to do so.
Fantastic. Thanks for that great intro, Joe.
Thanks, Shiz.
So let's dive into the 5 easy to use tips for, that's just a publication, my apologies, publication figures. But although, as Joe mentioned, they could definitely be used for presentations as well. And again, these are the sort of 5 things that we've tried to consolidate, based on these, I guess, more overarching concepts. So color, layout, clarity, fonts and labels, and contrast. You've probably heard us talk about 1 or 2 of these before in different contexts. So hopefully this will be more clear to you as to why they're important for publication figures in particular. So let's start with the first one. The idea of using complementary colors or sort of color picking based on the objects on your canvas. And the concept of complementary colors is pretty straightforward. So many of you have probably heard us talk about this, or maybe learned about it in grade school even. So complementary colors is just a fancy word for an opposite color on the color wheel. And it sounds very rudimentary, but even as designers in industry, we use this day in and day out because we know it's effective, and it makes picking colors kind of easy without having to think too much about whether things go together, whether they clash, whether they stand out against each other as well. So orange and blue are opposite colors or complementary colors. Same goes for yellow and purple. Red and green are opposite colors, kind of like the Christmas color combo, but we tend to sort of shy away from that a little bit because of our colorblind audience. We don't want them to lose out on that information. So I recommend using orange and blue, yellow and purple, and there's a lot of applications of that in science illustration already, probably without you even knowing it. So the sort of blue and orange combo really makes those orange elements in the foreground stand out, especially if they're kind of antagonistic concepts. So bacteria kind of on top of this pile of lymphocytes, and the lungs on top of this 3D human here. And even this kind of neuron on a purple background, you probably didn't notice that those were complementary colors, but something about it made those neurons really pop out of the screen. We usually, as a caveat, I do say to use background colors very sparingly. So I wouldn't recommend just going in and making a dark background for everything you're doing. But in this case, it just happens to work really well, and it's because they're complementary colors. Same goes for data sets. So if you can get away with using, I guess, monochromatic or variations of one color to denote one data set against maybe an opposing data set that's the opposite color, that also tends to be very effective. So instead of using, blue and green for two data sets that are almost very close in hue or color, I'd recommend going the extreme opposite spectrum and try to pick colors that are complementary or opposite. Of course, sometimes you can't get away with using the sort of rainbow of colors if you're representing many, many data points. But if you can get away with it, this is a nice way to really kind of strikingly make a balance between different opposing data sets. And here's a really beautiful image that I found in, it looks like it's the wrong link there at the bottom. I liked how they, in this case, use sort of that sort of bluish cool color against the reddish warm color. Now, these aren't necessarily opposite or complementary colors, but they're close enough that they kind of really give a nice range. In addition, they've actually carried through the red and the blue all the way throughout the data sets. And on first glance, it looks like it is, in fact, speaking about, for the most part, the same thing. So depending on how you're showing your data, if you stay consistent with the color palette that you're using, it makes audiences and editors it makes their job much easier to follow the story from beginning to end. I also like this example because they've really clearly labeled the alphanumerics or the sequence. So A, B, C, D, E, F, G is very clearly outlined. That might be the nature of the editors or the designers later on, maybe editing figures. Sometimes they will do that. But if they don't have a hand in it, I would make sure, right from the get-go to make sure that your sequences are properly labeled. The numbers or the letters are nice and big and bold. So it's easy to follow, again, that sequence from top to bottom or left to right. And here's another really beautiful example, actually from Dr. Zeppa himself. So maybe he can chime in here to describe a little bit about this figure.
Awesome. Thanks, Shiz. Yes, this is from a publication I co-authored back in 2014 in PLOS Pathogens. Big shout out to Dr. Catherine Casper, who was the primary lead author on that, and Dr. John McCormick, who actually was my mentor at the time, as well as he actually was the one to do a lot of this layout. So what I really like here, again, as Shiz mentioned from the previous slide, is kind of how they're nicely grouped together, A, B, C, and very easy to follow. The color scheme also follows a similar trend to the previous article, the kind of blue, red, white. I guess the one caveat here you could say is that you might actually think that the red, blue, and white kind of follow through. You'll actually note in panel A, the blue is the DAPI nucleus stain in the immunohistochemistry image, and the bacteria is red, kind of in the lower portion of that image. And the right, we're now denoting the colors actually as different strains. So the red is the wild type strain, and the blue is one that's lacking a certain genetic element. Then in the bottom, you actually note we're now using red and blue as a gradient for those different cytokines and chemokines. So I believe in the figure legend, it was nicely outlined, but just be careful that people don't draw the wrong conclusions when you're using the same color theme. But still, as long as you outline it in the figure legend, I think that's okay. And for those of you who are interested as well, the left images of the immunohistochemistry were formatted in actually Adobe Illustrator. This was pre-BioRender 2014. Top right is a prism graph, and bottom right is actually an online tool that people can use that allow you to create these nice matrices that is called Matrix 2 PNG. So again, I think we did a good job here with the layout, and actually I think the color does look nice, but I think you just got to be careful to actually describe it in your figure legends when the colors don't carry through in each one of your actual images.
Yeah, great point, Joe. Thank you. And sometimes you can't really get away from it because those color stains were kind of limited by what those offer. And like you said, I think you did a beautiful job. You and your co-authors did a beautiful job of laying out the overall design. Beautiful grid system, beautifully aligned. And then we'll kind of show you how to do that as well in BioRender using some of our grid line tools. So thanks, Joe. Great. On that note, I guess we're going with the red and blue theme here because this is another example. It wasn't intentional. But just thinking about how to match colors in your figure itself. Generally speaking, this is, I guess, slightly unrelated, but a little tidbit here is when we tell stories, we like to try to make sure that, again, even if you didn't know about the science, about what the labels said, if it was in a different language even, you should know right away coming to a figure that there are certain bad guys in the story and good guys. In this case, because the sort of tau protein tangles are perhaps a naturally occurring thing in the body, but in this case is causing the pathology. It's shown in red. Obviously in the body, it probably isn't red, but in this case, it's color-coded red, and it kind of immediately tells me, okay, that's kind of the bad guys in the stories or the antagonists. And then the protagonists or the good guys or the things that you want to save or the normal biology is actually this sort of microtubule. Of course, it's disintegrating in this case, but the blue versus the red kind of really tells that story. If it was the reverse, it would actually maybe look like the microtubule is kind of the bad guy or causing the pathology in this case. So just keep in mind kind of that warm, cool color combo. As well, if you want to tuck away some of your labels, as is showing here, by removing the arrows and then tucking the arrow closer to the object that it's labeling, that's actually a nice way to save space and also to really clearly identify what label goes with what object. Sometimes you'll get kind of a cobweb effect where there's labels and arrows and colors all over the place. It starts to get difficult to see what is labeling what. And then finally, the way that the arrow has been used in this case was a little bit confusing because it's the same arrowheads and the same shape as it's showing above that denote motion or this sort of cyclical pathway. You should really use a different type of arrow when you're labeling. And we actually have a label line in BioRender that you can use that will differentiate action from labels. Another kind of concept or way to color pick, I use the Columbia University Medical Center logo here just as going with that sort of blue theme. But of course, you can color pick from the TAMU logo, which is a beautiful sort of mahogany red color. All you would do is color pick, usually on your school website. It might even show the hex code of what your logo is. So this might apply a little bit more to posters, but if you do want to start to brand your images to color pick from your school or from your company, or maybe even from a microscopy slide, you want to color pick the color of the stain so that it looks similar. You can actually use this technique. So just grab the hex code. It's like a 6-digit alphanumeric, and then plug it into whatever software you're using, like an Illustrator or a BioRender, hopefully, and then it'll give you the exact color swatch. So just another kind of tidbit on how to unify your colors across figures. Okay, and then we'll go into a little more detail during the demo itself. Second tip that we'll kind of cover here is sort of layout. So the use of guidelines and or boxes is what we do internally to make sure that all of our, I guess, panels or figures are well aligned. And I'll tell you what that means in just a minute. And if you attended our previous webinar, we did touch on this, but quite, different applications again with publications. So for this particular image, it's really nicely aligned. I like showing this example because it's got really consistent padding and spacing, even in areas you don't expect. For example, within that right column, those lines are relatively even if you kind of draw these artificial grid lines on top. And again, as industry professionals, we really rely on guidelines to help us align text, pictures, headings, subheadings, even just leaving enough space around the edges so that you have some visual breathing room. So the take-home message here is try to use the guidelines or grid lines, they're called in some cases, built into tools that are available to you to align your objects better. There you go. And I think you've seen us show this before, but I like using the analogy of Pac-Man. So sometimes when you think about spacing out your paragraphs, it's hard to think, okay, is this parallel enough? If you can imagine Pac-Man kind of going up and between your sequences, or I guess your rows or columns, then you've done a good job of leaving consistent parallel padding. So this is just kind of a visual rule of thumb to make sure that you follow this kind of general rule. In this case, we've used a poster, but again, this applies to multi-panel figures for publications. Here's an example I believe you showed in the last webinar as well, but this really just drives the sort of message home of how to even take a relatively well laid out figure like this and really make it evenly spaced out. So this is actually a figure that we have in BioRender as a template that you all can go in and use today. All you have to do is, swap out some of the labels to make it more relevant for you. But before we published this, we actually made sure to maybe make it a little more aligned and consistent in the way that it's been composed. You'll see here, if we add boxes around each step, we started to notice that not only were they uneven in size, but uneven in its spacing between the boxes. So you don't have to be perfect in this, but this is just kind of a way, again, rule of thumb, to make sure that all of your steps are relatively even or at least symmetrical. So here's the after, where we kind of realigned all of the boxes. And if you, again, draw those artificial boxes, now it's sort of more consistent. I didn't put a box around number 4 because it would have been a little bit larger, but I wanted to show that roughly speaking, the top row is all the same size and the bottom row is all the same size. Again, they don't have to be the exact same size, but it's at least better than what we had before here, where they were all different sizes, all different distances from each other. And that does make quite a bit of a difference when you kind of toggle between the two. So just keep that in mind, kind of use either grid lines or invisible or even actual boxes around your steps to see, where they end and begin. And here's another example we also have probably shown before, but I think is a really good example of how to lay out sequences better. So maybe, if you're showing stepwise what you actually did in your experiment to come up with your results, you really want to make sure you follow traditional kind of conventional composition rules. So left to right, up to down, maybe it's cyclical, maybe there's a fork in the road. But in this case, it was kind of a combination of all of those things. So it goes from left to right, down, and then back, which is a little bit unconventional. So now we're reading right to left, and there's kind of this fork. So what we did was literally just did the same thing, followed the left to right rule, and then kind of put little boxes around each step. And then same thing with, kind of making those words and pictures consistent. We made sure that all the numbers were in a row, all the pictures were in a row consistently, and all the labels were in a row. So it might seem like a minor change, but keeping those steps consistent really helps alleviate the stress of the viewer and the reader and even editors when they're coming to see your figures. So I hope that's clear, the before and after. We went with a more of a Z composition here. I guess it's kind of linear, but it's kind of left to right and then switching back to the beginning like a typewriter and then going again left to right again. Okay. Here's another example I kind of took as a really well laid out, really sort of scientifically rigorous and interesting concepts. But I thought, why don't we just tweak it a little bit to show you what I mean by this sort of laying out boxes and better arranging multi-panel figures if you have a lot of data to show. So what I did was I, again, traced out most of the rough areas here. So A, B, C, I kind of put together because they're stacked, D, E, and F. I would say the reading order is a little bit difficult because I'm going from up to down and then right and then back to down again. But sometimes you can't get away with it. Again, as Joe mentioned, as long as it's very clearly labeled, you can probably get away with a little bit of an unconventional reading order. So what will we do in this case? We'd probably rearrange the boxes now to be more evenly spaced out and make sure we have padding on the edges. That is actually really important, to not put your letters or any kind of objects right to the edge of the page. Something about this just doesn't look professional. It makes such a difference when you leave a little bit of space on the edges. So here it is in the after state. Again, very, very minor changes, but having that alignment and spacing, and again, thinking of Pac-Man again, being able to go through all these margins relatively evenly is a good rule of thumb. And then here is that with the boxes removed. And I think I put the letters back in, there we go. So there was the before, and here's the after. So it just looks a little bit more professional, a little more cleaned up, and it looks like you've put a little bit more thought and care into sort of the overall layout. Okay, so that's the boxes rule, if you want to kind of take away one thing from this. And then going into number 3 here, clarity. So I think one good thing to think about is a lot of your images will be maybe photographs or data sets, a little bit difficult to digest on first glance. So to accompany that complex information with simple schematics is, I think, a really good idea in many cases. Of course, BioRender is a great solution for this as well. This is an example of a figure that we've created in the past with National Geographic. It was sort of a 12-page pull-out, kind of a centerfold, and obviously very, very complex. I think we did a good job of showing a multitude of species and their migration pathways all over the world. Really, really interesting story. But at the end of the day, we really wanted to show simplified schematics. So in this case, if you look at the bottom panel, a lot of this is just really simple vectors that kind of describe a complex story in very few pixels. In fact, if we zoom in a little bit, something to show kind of the life cycle of the eel, the European eel, we accompanied it with this schematic. If we had rendered this schematic in full resolution or full 3D or full textures and painting, it would have just been too much to consume. So sometimes the majority of your concepts can be shown in the simple schematic if you pair it, say, with the photograph or the data set. Same thing goes with this sort of shark inset here. We wanted to kind of show a zoom in of the kind of nerve fibers that are happening around the nose area of this great white shark. So this very simple schematic did the trick. We could have gone full 3D and painted this, but really we had to stop ourselves and say all we need is a schematic with labels, and that kind of orients the viewer to understand what's happening with the bigger data set or the more complex figure. It's a really great kind of trick we use in the real world. This is actually pulled from my thesis back in my grad school days at Hopkins. So this was talking about non-closure defects in organogenesis with the frizzled 1 and 2 gene with Dr. Jeremy Nathans, and really, really cool publication that I was able to be part of. Very figure heavy. So multi-panel, lots of data sets, lots of figures, photos, stains. And we thought, the best way to kind of tie this all together, because it actually was a very visual story. It was non-closure defects of the neural tube, of the ventricular septum, which is VSD, or ventricular septal defect for, being born with a hole in your heart, basically. Cleft palate. What else was there? I should know this. This was 10 years ago now, but multiple organs around the body. And what we did was we actually came up with more of a schematic to unify all of this. And I believe it ended up being the graphical abstract, or maybe at least one of the main images. And this is kind of what tied together the entire story. In this case, of course, I did this from scratch, but you could feasibly accomplish this through BioRender. Very, very basic, showing outlines of parts of the body where potentially this gene mutation could have an influence. But definitely more clear than showing, this number of data sets and showing that there's a comparison between them all, just because of the visualization modalities were so different. So this kind of showed across 8 organs the potential effects that this gene mutation could have. So again, comparing the schematic to heavy data sets is a really nice way to orient your viewer. Okay. Fourth thing here is fonts and labels. So this is actually really important to create hierarchy. Sometimes what we'll do is we'll lay out everything and then throw in some labels into the figure, not really thinking about what is the most important label and what is the least important. And this is really, really important, something that we scrutinize in the design world. So purely based on size and boldness, and even font type in some cases, forces your reader to read things from top to bottom, left to right. And this obviously is reading from top to bottom. But I bet you even if I rearranged these sentences in sort of vertical order, you'd probably read the boldest part first, even if we don't number it 1 to 3. So just to know that it's a very powerful tool to use font hierarchy to lead your viewer's eye through the paper or through the figure. In fact, a lot of successful infographics, now this is just a template, so take this with a grain of salt, but a lot of the times successful infographics are actually because the sequence number or the title of that sequence is the biggest and boldest, and everything is kind of really small and tucked away. So you'll notice this trend of really big numbers or really big titles. Here's a similar example. Obviously, this is a little more playful of a layout, and probably the audience is a bit more lay audience as opposed to a scientific audience. But the first thing you're going to read is the 1 to 7. And if you're curious or interested enough, you're probably going to read, maybe the details of each, city here that's described. But hierarchy of text is really the important thing. You probably read the title first because it was the biggest and boldest, and then the subtitles, and then perhaps the captions within each step. Okay, so this is an extreme example. Infographics tend to be really extreme, where you've got huge letters and then small text. But just to say that we can kind of take from this as lessons when applying it to science figures. Again, same thing here, largest text first, and you're probably going to read the smaller text later. Sorry, it's a little bit blurry here on my screen. And here's an example again of using hierarchy of text. So you probably didn't realize this, but in this example we showed earlier, the largest text is the header. And then the second largest is actually the title for each step. And then we make the description even a little bit smaller. So if you are allowed to do this variation of text size, if your publication calls for it or allows for it, I know some have strict rules about size of headers and descriptions and such, and font type. I would recommend trying to incorporate some hierarchy of text size in your figure labels. Great. I think this is also one of your figures, right, Joe?
Yeah, correct. So this is a figure from a recent Nature publication that I was a co-author in just at the beginning of the year. Big shout out to Dr. Trisha Dara, who is one of the ones who kind of brought all the data and all the figures together. Her and her group at the NIH with Dr. Mario Roederer and Bob Cedar. And of course, this was also done with me and my mentor at the University of Pittsburgh, Dr. Joanne Flynn, and our entire team. And what I really think that this does, actually, it wraps together a lot of the common themes that Shiz has been talking about. So you'll see kind of the largest bolded text are actually all of our figure insets, so you can kind of follow them along, A all the way through H. We also do a really good job of that compartmentalization. So everything's kind of a nice box that if you were to draw a square around it, they all fit very well together. The top right is a perfect example. A big question we get all the time in these publication figures is that, what do I do if my data doesn't fit beautifully into a box? What if it's a circle, or what if it has an odd number of panels or shapes? As you can see here, we had a couple groups that had the same number of scans. Those are PET CT images there, but a few of them didn't. So what we were able to do is, because the background in these were black, we were kind of able to generate a box behind them and fill that in with black. So it actually then turned into one large square that we could fit nicely into this image. Also going along with kind of our color format from before, now this is actually one circumstance where we were able to carry through the color to each one of our panels, insofar as each one of our treatment groups had the same color. So jumping back and forth between each panel, you knew that the red was the IV or intravenous group, etc. One of the last tips actually I wanted to give you that I've just realized might be helpful for some individuals was the bottom figures there were made in Prism. They're just your classic Prism plot. But what we actually noticed was one of our groups had a lot of animals that actually had 0 colony forming units or bacteria. So one of the cool things we did was, when we kind of formatted this on the x-axis, we just had a lot of circles kind of overlaying each other. And then what we realized is, if we actually created a grayscale in the bottom and gave these kind of fake numbers in the negatives, we were able to spread out and show how many of our animals actually were prevented from being infection in this article. So I think that's kind of another thing. If you have a lot of units that are stuck on the axis, in Prism, you can actually generate a negative into that axis and kind of fill it out a little bit nicer, so you can just demonstrate how many of your animals were protected, such as in this situation.
Really cool, elegant solution. Thanks, Joe. Yeah, I hope you can take a few tips from this, as well as how Joe and his team have integrated all the tips. And if you have questions about the whole publication process with Nature, you can probably ask him in the chat. I'm sure he'd be curious to hear. Great. So final tip here before we run into a quick demo, I think we're doing pretty good on time, is the idea of contrast. So this is something that we talk about a lot. I think it's probably, at the end of the day, one of the most important, if not the most important tip we can leave you with. And that is really to ensure that you have enough contrast in your figures for many, many reasons. Here's kind of an example again of showing the idea of color contrast and how it can kind of come back to bite you if it's not done well. 2 images that if we switch to black and white or preview and grayscale, one kind of completely disappears because they're very close in color value. So just a fancy name for the darkness or lightness of a figure and its elements. So really the rule of thumb or the take-home message here, just to repeat as we always do, is to not stack parts of an image that are close in color value. And part 2 for this is to even use the kind of preview in grayscale mode that a lot of apps have, including BioRender. So don't stack parts of an image that are close in color value or darkness or lightness. Make sure to stack more light on dark or dark on light, as is shown with this horizontal circle here, or rather diagonal circle, sorry. Here's that preview in grayscale trick that I was mentioning in BioRender. So you see here, going back to that example with the stabilizing tau proteins. By previewing in grayscale, it actually knocks out color as a variable. So now we can see that indeed those objects are way too close in color value or darkness or lightness to the background elements. So we can kind of go in, select all same object, change the color of that icon, And then now there's enough contrast, and even the color we've selected is actually telling of what is the quote unquote, again, bad guy in the story. So that's the preview and grayscale mode. I think if you're using Adobe Illustrator, they have a grayscale mode as well. Same with Photoshop. Really good rule of thumb. Okay, that's not the end of the demo. We'll still have a few minutes here for a quick kind of example in BioRender, but I'll pause for a second to see if there's any burning questions that we haven't answered in the chat. Sounds like we're doing okay, Fran and Lucas, Joe?
Doing great.
All right. Fantastic. Thank you. Here's that portal that Fran did talk about at the beginning. If you haven't had access to BioRender already, it's again app.biorender.com/portal/TAMU. I'm going to sign in with my generic portal because I am not part of Texas A&M University. So this is a generic portal that I would have logged in from. So I'm going to go ahead and do that. And again, for those that aren't familiar with BioRender, really quick kind of overview of the user interface or UI. These are my illustrations on the bottom here. Actually, I've probably got hundreds by now, so I won't scroll to the bottom. But I like to keep my images organized by folder all on the left-hand panel. I can actually, sort my images by putting it into the correct folder. I can create new folders. I can create images and add it to my team gallery. So I suspect some of you might be working with the same individuals on any given day, maybe classmates or lab mates. If you're a PI, maybe you have students in your lab that you'd like to share figures with. You can do that through the team gallery. We also have templates. So again, if the idea of starting with a new application or starting from scratch is intimidating, we have some really nice starting points from which you can make a figure. So even if it's not directly related to what you're working on, if you like the way that our artists have laid this out, you can actually go in, swap out the labels, swap the colors out, however you like to edit the figures. All organized by subcategory roughly, but you can also search by keyword. So if I type in neuro, you can find some neuro-related templates. And again, all fully editable. If you work with mouse models, immunology. So go ahead and search our templates tab and see if there's anything there that relates to your research. So going back to the gallery here, I'm actually going to go through and select my arrows tutorial. So this is one thing that I think is really important when going through and working on images that, again, have multiple different arrow types. Here we go. Lines and arrows. So we did talk about staying consistent with the types of arrows you use. And this is something that I would highly recommend when you're going through making your figures. And also if you have a lot of competing elements on your screen. So I'm just going to quickly go through 6 different types of arrows that you can incorporate into your publication figure. Because again, sometimes it's the last thing you think about, but it actually should be incorporated as you go through in the figure making process. So arrows and labels should be considered as part of the figure itself, not sort of a add-on that you slap on at the end. So what I'm using here is our trusty circular arrow that we have in BioRender. It is a perfectly 360 arrow that you can use to create cyclical motion, plasmids, any other applications you can think of that involve perfect 360 circles like this. Maybe I'll rotate it and even snip it down the middle to create a cut. This is a really cool trick. And in some cases, if I want to show that it's looping over and over, I'll even add a fade to the tail like so. There's the before and after. And then going back to the idea of really driving the message home of what is kind of the good guy and the bad guy in the story, I've made this into an inhibitor line just by clicking the arrowhead types here. And then maybe I'll even change the color of it to show that, one is indeed maybe a good thing, one is a bad thing. There we go. And I'm going to just view in grayscale to make sure that that's pretty close in color contrast, the two arrows, and still stands out, so that's good. Great. Another thing that I would recommend is differentiating your label arrows from action. So these are the arrow labels that I was talking about earlier that I'd recommend. It's this line with this dot at the end, a little bit different looking from the lines with the arrowheads, which again show a little bit more about motion or directionality. This is more so labeling an object. So if I zoom in a little bit, and I'm gonna just hit Option drag for those on a Mac. I think it's Alt drag for PC if I'm not mistaken. Really great way to label things without confusing your viewer if you again have objects or pictures that you have to add multiple labels to. Something about having this dot at the end really does help clarify where that line ends and begins. If I were to take that off, it's a little harder to see, where is it that line kind of lands. If I was zoomed out a little bit here, you can see. So if I were to add that dot, it just makes a bit of a difference. I know I'm kind of belaboring the point here, but it is a trick that we like to use in the design industry. Okay, so that's labels. Sectioning is another way of using lines to your advantage. So dotted lines can be used for this case. Now, a lot of us like to use dotted lines for many reasons. It does also represent a pretty different concept. So dotted lines could mean, cut here or fold here or remove here. It's kind of a past or future event. So something that is or has happened. And something about it being dotted just shows that it's, not a for sure thing. Or in fact, it could just show sections like this. Sometimes in some cases, if it's a circular dotted line, it could again show that this is an incision mark if you want to remove a tumor in some cases. So just know that, again, showing different types of lines can represent very different things. In this case, the contrast is not that great, so I'm actually going to make the lines white. So change the line color to white. There we go. So just to show you also to highlight the different, line options. Oop, wrong line. But yeah, you can see that you can create any line into a dotted line in BioRender if you need. Okay. Showing volume. So differences in volume can also be denoted by the thickness of the arrow. This is very obvious, perhaps sounding or subtle, but if I show two different arrows, of course it's going to show directionality, but you can take this opportunity to even show volume. So just by creating a thicker or thinner arrow, you're automatically communicating that the volume or the power can be accentuated by the thickness of the arrow itself. So arrows can actually be part of the illustration, not just as a label or an afterthought. So sometimes I'll take this a step further and actually color pick. I think we have a color picker here. So here I'll pick my little eyedropper and color pick the text. So now it matches exactly the text in the line. The portal vein, that font is starting to look a little faint, so maybe I'll make it a bit darker to be a dark blue. And I'm gonna want this arrow to be that same color. So let's see, color of the line, I'm gonna color pick. And if you look at the top left of my screen, it's just picking up, the color regions that I'm overlaying. So there we go. So right away, it's showing that portal blood or venous blood is coming through the portal vein from the intestines, and the bile is going down towards the intestines, like so. So again, communicating volume just by changing the thickness of the arrow. Okay. If you do want to take advantage of the full suite of tools here in the color palette, with the Texas A&M subscription, you actually get access to even our gradient tool. So if you don't already know, we actually have gradients as a color option. It's a little bit tucked away. So I can't quite make this line a gradient, but if I did want to make an arrow gradient, we get that question a lot, is I could do it this way, where I make a shape. And sometimes you want to show, for example, differences in concentration or temperature or pH. So I'm going to remove the border color and then grab the gradient fill, which is next to the solid fill here. And we do have a couple of options as far as pre-made color gradients. And you can get pretty granular with, you know, sliding this back and forth. If you're savvy with Adobe products, you'll know that they also have something similar. But this is kind of at your fingertips and available for you to use if you need. And of course, you can switch the direction of the temperature or the concentration or the colors like so. Okay, so just know that that's an option for you if, you know, for some reason you need to show that. All right, last few here is transcription. Of course, very simple, but very communicative is our inhibitor lines and our action arrows or promoter arrows. So I can easily switch from an inhibitor line to a regular arrowhead like so. And again, you're going to want to use a perfect 90-degree angle for this. And back to inhibitor. So really easy to swap back and forth. Movement is the last thing I'll talk about here. If you have kind of a spray of cytokines or dots or peptides, what have you. Sometimes if you want to show that things are kind of flying in or moving around, we like to show that with this sort of faded, curved arrow. Here we go, something like this. Again, very different from that perfect cyclical arrow, different from, you know, the transcription arrows or the straight arrows. These curved, almost tapered, faded arrows represent movement. And you can actually start to curve this in funky directions. If that's important for your story, you know, don't use unnecessary accessories, but if it's important, you can actually use the arrow in this way and make it really flexible as to, you know, what you're showing. But that faded tail really helps with showing movement, kind of like the Nike logo. It's like that swoosh motion. It kind of makes it look like things are moving as opposed to starting and stopping pretty statically. Okay. And then if you didn't notice, one little tidbit here, design trick, is things flying into the distance tend to get smaller and actually start to look more faded. So that's what we do sometimes as a trick in industry, is we go from 100% opacity to about maybe 50%, because naturally things that fly into the distance do get this kind of hazy effect. So it's an optical illusion that we were creating 3D space here by decreasing the size of those molecules and actually decreasing the opacity using this opacity slider. So I'm throwing a lot of tips at you here, so I hope I'm not losing you. But I thought it'd be worth kind of covering these 6 arrowhead types for this particular webinar, because I think we see that a lot as a mistake in going through making figures. Any questions we have from the attendees here? Still got a very full room, so thank you for your attention. Hopefully you're finding this helpful. Looks like we're doing okay. Oops, I'm gonna save that. And all for those of you who are new to BioRender, hopefully you've picked up a thing or two now going through the tips and tricks. I think one thing that, again, we'll cover going through the contrast issue. Now, we showed this in the last webinar in a speed through GIF, but here it is a little bit more slowly. If I were to use this figure as an example of something that's pretty low contrast, I'm going to, again, go to preview and grayscale, and the DNA almost completely disappeared. I see a question in the chat there of how do we change the color of the icons? Great question. So in this case, I'm going to change the color of the DNA to be probably a little bit darker. You can see already in situ, it's changing the color as I scroll, so that's nice. I would either have to go very bright like this or very dark. That's just the nature of working with cells that have dark nuclei, basically all of them. So sometimes what I'll do is, again, knock back the opacity. Don't be afraid to play with this opacity slider to make your foreground element stand out. Or, as was asked in the panel or in the chat there, I can actually change the color of the icon. So it depends on the type of icon that you want to change the color for, because the way you do it is a little bit different depending on the object type. This, in our case, is an icon, or what we call an icon. So you can change the color like this holistically, like so. And even this cancer cell, I can change to something else, like this. In some cases, let's see here. For example, this arrow, how to change this arrow color you saw earlier is actually a little bit more granular. So I get a whole palette because it's more of a dynamic shape versus an icon. So those are the two differences. In some cases, and not to confuse you, but some of our icons are actually made with shapes. So you can get pretty granular with how you change the colors of some icons. So comparing these two CAR proteins, they look the same, but they're not actually the same because one is made with shapes, one is made from our artists using a vector program. So the difference is really in the color of the bounding box usually. This one is purple to denote that you can double click to edit. So I'll double click, and now I can change these to an infinite number of colors. So if I move it out of the way, I can change it to maybe blue, yellow, or any color of the rainbow. And I can even, remove maybe one or two of these domains if that doesn't make sense for my story. I can change this backbone. Whereas in this case, it's kind of a quote unquote flattened icon, even though it is still vector. The whole thing is going to change as I scroll through these almost different, shirt colors in a way. So I hope that makes sense. Those are the two ways you can change colors. I hope that answers your question. Okay. So let's check for grayscale here to make sure there's enough contrast. It looks pretty good. Nothing's getting lost. I think this label's looking a little light. Maybe the arrow's a little light too, but maybe that's okay for my purposes. And yeah, looking pretty good. I like the way that the text hierarchy is placed. So the T cell and cancer cell labels are the biggest. And then I look down to the other label text as well. And then the main message is, of course, from left to right, as opposed to going from right to left. Okay. And then, if you want to export this and add it to your PowerPoint slide or import it into your Word doc to submit, depending on how you submit to a journal, I'd recommend exporting as the highest resolution we have here, 300 dpi, which all the journals should accept. We actually work very closely with a lot of journal editors to make sure that we do follow protocols for journal guidelines. And I'm going to go ahead and throw this into my desktop. And then back to our slide deck here and insert the image. This is just to show that you could incorporate it into a slide deck, publication, what have you. So it comes in very, very sharp and clean. This is 300 dpi, so if you zoom in, you won't see any pixels for a long time. And you could have actually exported it with a transparent background, so all of the white will be see-through if you export as a PNG. So lots of options there, again, if you want to export it for different purposes. So we're coming up to 12 o'clock your time, I think, 1 o'clock my time. So I think we'll wrap up here. I hope this was helpful. And big thank you to Alex Trott, Associate Director of Rodent Preclinical Phenotyping Core. for helping us organize today's session. It sounds like you do a lot for the researchers and folks there. So thank you so much for taking the time out of your busy schedule. And everyone here who joined the webinar, hopefully you took away a thing or two. We're actually going to be running 101 sessions, so BioRender Basics, every Wednesday, and that's open to the public. I believe you can subscribe or sign up through biorender.com/webinar, and it actually shows all of our themed webinars. And then I'll also leave you with a really quick call out to join our graphical abstracts contest. It's really exciting. We're only running it for another 3 weeks. So if you have a picture or an illustration you've made in BioRender, you can actually submit it to our graphical abstract contest. There's actual real cash prizes. So really fun kind of initiative we've done here. We've taken all of our conference budget now that we're not actually physically traveling anymore, and we're offering it to you as a cash prize to your figures. So a lot of these images made by your colleagues are following a lot of the rules that we talk about as far as design principles. So you can take some inspiration from the gallery there. And we've actually recently launched our early bird prize yesterday. So look at the top examples that use really good, effective, labeling, spacing. This one's a great example. So go ahead and do that. I think it's a really fun initiative, and love to see what you submit. And yeah, thank you so much for your attention.
Thank you everyone for joining. And as Shiz mentioned, I did write the portal for Texas A&M, and you can access your free premium account through that link.
Thanks, everyone.
Thanks, everyone.
Have a great rest of your day.
Have a great weekend.